Good evening, and thank you. I'm glad I was able to be here today. Carmell is got some fantastic technology to improve the healing process. That's significant because healing time and effectiveness cost money, and we have technology that not only heals faster but also reduces complications. We're on a path to open a Series B of about $1.5 million to $2 million on a way to a Series C that will be about a $10 million raise that will take us to exit. Our planned exit is through an acquisition to a multinational company. So what we have is we have technology that is derived from the source material of pooled plasma. This is collected from the blood collection process. The red blood cells are used for transfusion. That's where your typing comes from. There's no typing associated with plasma, but the plasma has all the good stuff in it. It has the platelets, the growth factors, all stimulatory, inhibitory factors all contained. We keep it all in that form. We do not fractionate it. We process it. The scientists at Carnegie Mellon have been able to find a way to do three important things. One is form. They've been able to create three-dimensional shapes of a putty, a paste. Those are our regenerative products that we currently have in development, but we also have concepts for a sheet, a ribbon, a screw, and other forms. The second thing they did was able to control the degradation rate. The other problems that have been associated with trying to deliver growth factors to injured sites is that it washes away in 24 hours. This we can program to have it there for 2 weeks, or in the case of bone healing, you want it there for 2 months. The third thing is after we are able to do all this, it's still bioactive, meaning the growth factors are still present and they're delivered locally over the period of time that the body needs it for healing. So these three things are very significant. There are no other competitors that are using allogenic plasma, platelet-rich plasma for the purposes of healing. The company was started in Pittsburgh out of Carnegie Mellon. That's what Carmell stands for. We have our current investors of a combination of both angel as well as venture capital. We've already been through a Series A, and we've got 2 products in development. So when I arrived, I switched the company from a research-oriented company to development. Now we have a very strong focus on bringing these 2 products through the regulatory process for the purpose of US approval. We are a clinical development company. Our exit very clearly will be at the time we gain our US approval. So in order to do this, I needed to build a significant team. I worked for J&J for 13 years in senior management positions in the largest, most important drug device and biologic device combination products that they had. Dr. James Hart retired from J&J 2 years ago as the chief medical officer for J&J Global Surgery and is the chief medical officer for Carmell. Dr. Toby Silverman was the branch chief for CBER, which is the designated branch for us. She was the reviewer for the first BLA that Dr. Hart and I got at Ethicon Biosurgery called Evacell, a fibrin sealant, and then she left and went to industry and she became the consultant on the second one we did, which Everest, a fibrin pad. She now is the consultant for Carmell. Donna Godwood retired from J&J in April of this year as the chief quality officer for all of J&J's medical device and diagnostic sector. As Susan Morano said, that's a $30 billion business. She's now our chief quality officer. And Dr. Stephanie Kladakis is a VP of R&D. She moved over from Cohera Medical and has significant experience of taking products from prototype to FDA approval. So we have very, being an academic startup, they did a terrific job with asking a lot of questions and going to animals and getting animal data. We have substantial animal data, terrific biocompatibility data, but more importantly, we have human data. We have done a pilot study successfully. I'll share the results with you, but statistical significant reduction in the time for bone healing, as well as statistical significant reduction in infection rates with a very attractive p-value on a small study. So we're going to start with long bone fracture. That's where our data are in the pilot study. That's our first indication, but this is clearly a platform play. The regenerative putty will be used for orthopedics. We'll move into spine where we'll mix it with DBM, demineralized bone matrix, to extend that and use less DBM, but also enhance it. In the rat studies we've seen so far, we grow stronger bone faster when you mix it with our putty, and then as well as joints. 18 issued patents, another outcome of the academic setting. They did a terrific job of working the patent portfolio over the years. We are a BLA. This is not a drug. It's not through CDER. It's through CBER. The clinical program looks a lot like a PMA. in which we have designed our clinical study, and we have 200-patient study that we'll be conducting for the purposes of approval. What's different than a PMA is the CMC, Chemicals, Controls, and Manufacturing, and we feel very good about our potency assay that will demonstrate the biologic is active after the manufacturing process. So here's the product portfolio. First product is for a putty. And then be indication sequencing. This will be for bone. Second product is a paste. This is for soft tissue. Think about surgical site infection, simply applying the paste at the time of closure and having it release the growth factors, have VEGF, angiogenesis to enhance the healing process and reduce infections. The first indication is the long bone fracture. This is a tibia fracture coming through the skin. Automobile accidents, very significant healing challenges associated with that. Typically a 9 to 12 month process to heal. This technology is going to be extremely cost effective. We envision about $600 a procedure covered under the DRG. It only costs about $30 to $60 a procedure to make, so very high gross profit margins on that. The market size for these are all very large in themselves. For long bone fractures, 1.3 million of those a year in the U.S. times $600. You can see the significant sizes of the different markets for the putty and the paste, and these are the only two that are in product development, but other concepts would be available as well. Here are some of those concepts. We have molded a hard screw. There are bioabsorbable screws today, but none that release growth factors when they absorb. We have sheets and ribbons. And then we also have done drug delivery with an antibiotic and had a very successful rabbit model doing that. The study that we conducted was conducted in South Africa, 30 patients, 20 treatment, 10 control. The control had standard of care, which are going to be plates and screws. The treatment arm was standard of care plus the putty. The putty is room temperature, ready to use off the shelf. There's no mixing or prepping. You simply apply it to the bone fragments into the fracture line. And what we saw as a result is 4 times fewer infection, twice as fast bone healing, and 4 times faster wound healing. When you measure the strictest definition of healing at 180 days, these typically take 9 to 12 months to heal. So at 6 months, we're accelerating healing. We had, under the strictest definition, 36% of the patients were healed. For the most complicated patients, which are called 3A and 3B, 0 in the control. And then the infection reduction was extremely significant statistically. We're raising a Series B. This will allow us to go through the FDA process to get our IND so that we can conduct our study, get the IRBs through, the contracts up, 12 to 15 sites. Then we'll do a very cost-effective clinical study that will follow the standard course of care. 3, 6, 12 months of x-rays, AP and lateral, so we won't have to pay for that, so our clinical trial will be extremely efficient. The model that I used was I looked at the Silicon Valley Bank publication early this year. We're extremely capital efficient. Once we get to that point of enrolling, we will have spent $9 million. The standard is typically about $22 million. We think we can get to a BLA approval. I'm using PMA as a benchmark. For about $20 million versus $60 million standard, and the deal size for this type of company or technology is usually about $300 million. So that'd be a 15x return. That's a thematic kind of representation of value as we hit our endpoints. The most relevant would be the database lock, where we would do the 200-patient study and repeat the results that we saw in the pilot study. So then you have 2 human studies confirming treatment effect and validating the technology. That would be the first exit point. The second exit point would be the BLA approval. We are in discussions with Blue Tree Allied Angels and Keiretsu Forum Mid-Atlantic co-leads as it relates to a term sheet. And pre-money is not determined yet. These are the last conversations we had. This is very fluid as I've attended this meeting. The due diligence is complete. The due diligence report is available from Blue Tree. And we're looking at a post-money of $12 million. Thank you very much.